starting with the chapter biological classification. For any chapter we need to know the framework, what are we learning and how much we need to learn for the exam, and how is it connected with the other chapters as well. So, biological classification very simple ra. First you have to learn on the systems of classification. Okay. Like for example, we have 2 kingdom system, 3, 4, 5, 6 kingdom systems of classification. So, first on what we have to learn? Kingdom systems of classification. Kingdom systems of classification. Where we learn in detail of what are the criteria considered for each system of classification, who proposed, and what is the new kingdom introduced for each system of classification. Likewise, what we will learn? 2 kingdom, 3 kingdom, 4, 5, 6 kingdom system of classification, of which the widely accepted system of classification is what? 5 kingdom system of classification. This is widely accepted. Why? Because in 5 kingdom system of classification, phylogenetic relationships or evolutionary relationships are considered, right? And this 5 kingdom system of classification, if you see, what are the 5 kingdoms that are there here? Monera, Protista, Fungi, Plantae and Animalia. These are the 5 kingdoms that are there. What is the new kingdom introduced in 5 kingdom system of classification is fungi. Fungi is the new kingdom which is introduced, right? After learning this, every kingdom we will be learning in detail. After noting on this, we will learn on those organisms which are not included under 5 kingdom system of classification. What are those organisms? Virus, Viroids, prions, Lichens, right. So, virus, Viroids, prions, Lichens, these are not included under 5 kingdom system of classification. About these organisms we will learn at the end, okay. And plant kingdom, few of the intro or description we will learn, animal kingdom also we will learn, and both of them we have 2 separate chapters, animal kingdom and plant kingdom in continuation. Got it right everybody? Yeah. So, shall we start with kingdom systems of classification? Everybody got an idea on what we are learning on this chapter, right? So, first these are the fundamental insights we have discussed. First, scientific method of classification is proposed by whom? Aristotle is the scientist who proposed first scientific method of classification, where he generally classified the living organisms into what? Plants and animals. He just normally classified the living organisms into plants and animals. After that, he has looked into the morphological characters, the phenotypic characters which are present. So, based on the gross morphological features or simple morphological characters, he has classified the plants into what? Herbs, shrubs, trees. In simple terms, based on length of the plant also you can say, okay. And animals based on the presence of red blood cells and absence of red blood cells, okay. This absence of red blood cells, anemia, it is addressed as anemia, and presence, anemia. So, this is the basic classification which is proposed by whom? Aristotle. So, who proposed the first scientific method of classification ma? Aristotle, right. Clear till here no? Now, we will move on to 2 kingdom system of classification. 2 kingdom system of classification is proposed by Carolus Linnaeus, okay. 2 kingdom system is proposed by Carolus Linnaeus, wherein many criteria are considered by him. Linnaeus has considered many criteria like cell wall, mode of nutrition, an organism's response to external stimuli, locomotion, contractile vacuoles, all these are the what? Criteria of 2 kingdom system of classification. But amongst this, one criteria is the main criteria, that is what? Cell wall. This you have to remember the most, because as he has given the first priority to cell wall, that has given many exceptions to two kingdom system of classification. Got it right, ma? So, what is the main criteria, major criteria for two kingdom system of classification? Cell wall, based on which all the organisms are classified: prokaryotes, eukaryotes, unicellular, multicellular organisms, autotrophic and heterotrophic organisms, all are classified on what criteria? First criteria is cell wall. Likewise, bacteria has cell wall or not? Yes. Algae has cell wall or not? Fungi has cell wall or not? Everything has dumped into what? Kingdom Plantae. This we have discussed in Cell the Unit of Life also, when we are discussing on cell wall, if you remember. Okay. So, here first is what? Cell wall in Plantae is present, in Animalia it is absent. Mode of nutrition is autotrophic here, here it is heterotrophic. Response to external stimuli by plants is slow. Response to external stimuli is fast here. Locomotion is absent in plant cells, it is present in animals. Contractile system is absent in plant cells, present in animals. Examples, if you see: bacteria placed under Plantae, algae placed under Plantae, fungi, higher plants, all are placed under Plantae. Whereas protozoans, vertebrates, invertebrates are placed under Animalia. So, as prokaryotes are along with eukaryotes, autotrophs are along with heterotrophs, is it like a dump yard, right? Okay, everything is dumped into a single place. So much of mess here. Okay, so that has given out so many exceptions. Likewise, if you see the drawbacks of two kingdom system of classification, there is a small paragraph in your NCERT below that table of five kingdom system of classification, okay, wherein he compares between two kingdom system and five kingdom system of classification, where he has given few examples. Yes, you can take one. Yeah, so where he has given few examples, okay, wherein prokaryotes are placed along with eukaryotes, he said. The example given here is blue-green algae. You tell me, blue-green algae is a prokaryote or eukaryote? Why? How? What is the criteria that you are considering and keeping it as prokaryote? And second question for you people, blue-green algae is autotrophic or heterotrophic? Huh? Autotrophic or heterotrophic? Autotrophic. Why? How? Presence of what pigments? Chlorophyll pigments. Where are they present? Hey, we have discussed, right? We have discussed, right, in Cell the Unit of Life, when we are discussing with prokaryotic cell and all. Okay. So, see here. Yeah. So, if I have to draw nostoc, in between heterosis. So, it is surrounded by a gelatinous sheet like this. This PDFs will be provided in the group, ma, need not to worry, focus on the class, ma. How much ever you can take, you take that much, rest you focus. PDFs will be sent in the group. Listen, so when we are reading about mesosomes in the prokaryotic cell, in Cell the Unit of Life, we have seen chromatophores are present in blue-green algae, chromatophores are present in cyanobacteria, where they are adjoined to the cell membrane. Okay. So, these chromatophores which are adjoined to the cell membrane, they will contain what? Pigments. Okay. These pigments like chlorophyll, carotenoids, they will be conducting what? Photosynthesis. Likewise, prokaryotic bacteria, blue-green algae, and it is a prokaryote because it does not have a membrane bound nucleus. Okay. Is placed along with eukaryotes like algae and bryophytes. Second exception, remember the examples, very important. Unicellular organisms like Chlamydomonas are placed along with multicellular organisms like Spirogyra. Both of them are algae only. You can take few more unicellular organisms like spirulina, chlorella, chlamydomonas, all these are what? Unicellular organisms. Whereas Pyrogyra, Ulothrix, chlamydomonas is filamentous green algae. Okay? Chlamydomonas, along with that you can also take chlorella, spirulina, along with this you can also take Ulothrix, chlamydomonas. Okay, and photosynthetic organisms like green algae or higher plants are placed along with non-photosynthetic or heterotrophs like fungi. So, what are these exceptions belonging to two-kingdom system of classification? Take down. Done? Next, three-kingdom system of classification. So, what are the two kingdoms we have learnt in two-kingdom system? Plantae and Animalia. Can you give me the exceptions from your side? Prokaryotes placed along with eukaryotes. Example, blue-green algae. Eukaryotes you can take anything, algae, bryophytes, pterido, anything. Second exception, unicellular placed along with multicellular. Unicellular exceptions, chlamydomonas, chlorella, spirulina, and multicellular like Spirogyra, chlamydomonas, Ulothrix, right? And next, photosynthetic placed along with non-photosynthetic. Photosynthetic you can take any of the divisions of plant kingdom, non-photosynthetic fungi is the example. Remembered? Now, three-kingdom system of classification. From here you have to observe who proposed and what is the new kingdom introduced. That much you remember enough from. Who proposed this? Ernst Haeckel proposed three-kingdom system of classification. And what is the new kingdom he has introduced? Protista. So, in five-kingdom system of classification, listen first, listen. In five-kingdom system of classification, Protista will contain all the unicellular organism. Okay? Unicellular organisms, plant-like, animal-like, fungi-like, unicellular protists you will have. But here Ernst Haeckel, what did he do is, he has placed all the unicellular organisms, be it prokaryotic, eukaryotic. In five kingdom you will see all the unicellular eukaryotes. But in three kingdom, when this kingdom was introduced, Protista, what did Ernst Haeckel do? He has placed all the unicellular organisms into the kingdom Protista. Okay? So, who proposed three kingdom? Ernst Haeckel. What is the new kingdom introduced? Protista. What are the organisms placed under this kingdom? Unicellular organisms. Right? Now, under four-kingdom system of classification, it is the scientist Copeland. Who is the scientist, ma? Copeland. He has introduced the kingdom Monera. So, now under three-kingdom system of classification, in Protista, all the unicellular organisms were placed, no? From that prokaryotes were taken and placed under Monera. Okay? So, in Monera, which is the new kingdom introduced under 4-kingdom system of classification, what are the organisms include? Prokaryotic organisms. Right, clear till here? Once let us recall. Who scientific method of classification proposed by Aristotle? 2 kingdom, Carolus Linnaeus. What are the kingdoms introduced? Plantae, Animalia. What is the major criteria? Cell wall. Exceptions you remember. 3 kingdom system of classification proposed by Ernst Haeckel. What is the new kingdom introduced? What are the organisms placed? Unicellular organisms. 4 kingdom system, co-plant. New kingdom introduced, Monera. Organisms placed, prokaryotic organisms. Now, our 5 kingdom system of classification, which is proposed by R.H. Whittaker. This is proposed by R.H. Whittaker, wherein 5 major criteria are considered. Cell structure, first is cell structure, thallus or body organization, mode of nutrition, reproduction and phylogenetic relationships. Remember this statement, amongst all the kingdom systems of classification, 5 kingdom system is widely accepted because they have considered what criteria? Phylogenetic relationships. Okay, they have considered what? Phylogenetic relationships. And in the table which is given here, okay, in your NCERT, many students will ask me the doubt, ma'am, the 5 criteria which are mentioned here are different and the 5 criteria which are given below the table are different. What we have to consider? You have to consider these 5 criteria which are considered for 5 kingdom system of classification, but of which in this table, cell type, cell wall, nuclear membrane, all these 3 will come under cell structure only, no? Yes or no? Cell structure, cell type, type of the cell wall and nuclear membrane, all the 3 comes under what? Cell structure. Body or thallus organization, right? And third criteria is what? Mode of nutrition. Reproduction and phylogenetic relationships are not given in this table. Okay? So, whenever in your objective about 5 criteria the question is asked, what you have to choose? Cell structure, body or thallus organization, mode of nutrition, reproduction, phylogenetic relationships. Now, one question for you. Cell wall is the major criteria for 2 kingdom system of classification. What is the major criteria for 5 kingdom system of classification? What is the major criteria for 5 kingdom system of classification? Mode of nutrition is the major criteria for 5 kingdom system of classification. I will tell you why. When we are comparing, I will tell you why. Okay? The major criteria for 5 kingdom system is mode of nutrition, and the system is widely accepted because of phylogenetic relationships. Okay, done. So, here it is like evolutionary tree. Monera, which is the first kingdom of 5 kingdom system of classification, includes unicellular prokaryotic organism. Second is Protista. Protista is the common ancestor for Plantae, Animalia and Fungi. This is the common ancestor. And this is the connecting link between prokaryotes and eukaryotes. Right, ma? All the statements which I am saying, please print and fix it in your mind, because those are most repeated statements or terms used. So, which kingdom amongst these 5 is the connecting link between prokaryotes and eukaryotes? Protista. Which kingdom has all the common ancestors for these 3 kingdoms? Plantae, Animalia, Fungi? Protista. Okay? Done? Got an idea, no? Now, let us examine this table and have a comprehensive analysis. So, first is what ma? Cell type. The cell type of Monera is prokaryotic, rest all will be eukaryotic. Cell wall, you tell me, the cell wall of Monera is composed of what? Peptidoglycan. Peptido, it contains of amino acid, glycan, it has glucose units in the form of NAG, N-acetyl glucosamine units. So, it has both amino acids and polysaccharides. Done with the cell wall of Monera. Protistans, they are of three types as we discussed. We have plant-like, animal-like, fungi-like. That is how it is forming the common ancestor for the three divisions, right? So, here they are present only in plant-like protists. Cell wall is present only in plant-like protists. That is how he used present in some. You can keep plant-like. Okay. Next, fungi. The cell wall present in fungi is what? Chitin, which has what units again? NAG units. This is also known as what? Fungal cellulose. Plant cell wall is composed of cellulose, hemicellulose, pectins, proteins, but most of the composition is with what? Cellulose. And cell wall is absent in animal cells. Got it? This is about cell wall. Then a nuclear membrane is absent only in prokaryotes, present in all of them, because all of them are eukaryotic. Next, body organization. Look at the hierarchy of body organization. It is cellular here in Monera, cellular in Protista also, because unicellular prokaryotes, eukaryotes are there, right? And in fungi, if you see, this is multicellular loose tissue type of organization. Multicellular loose tissue type of organization. Tissue and organ level of organization in Plantae, tissue, organ and organ system level of organization in Animalia. Remember the hierarchy, important. Last is what ma? Mode of nutrition. It is the most important criteria. You look at the comparison, then you will get to know. In Monera, you see ma, whenever you get the question like highest metabolic diversity is exhibited by which kingdom from 5 kingdom system of classification, so your answer should be Monera, because metabolic diversity means what will come? Nutrition will come, respiration will come. Highest nutritional diversity, you can see autotrophic monerans, autotrophic bacteria, heterotrophic bacteria. In autotrophic also you can see photosynthetic autotrophs, chemosynthetic autotrophs. Heterotrophs you can see three types: parasitic, symbiotic, saprophytic. So, highest nutritional diversity. When we talk about respiration, we have aerobic bacteria, anaerobic bacteria. Again in that we have facultative aerobes, facultative anaerobes, obligate aerobes, obligate anaerobes. So, highest metabolic diversity, your answer should be Monera. Highest nutritional diversity, respiration, Monera. So, that is with this kingdom. Next, autotrophic and heterotrophic both are exhibited by Protista. Plant-like protists will exhibit what nutrition? Autotrophic nutrition. Fungi and animal-like protists, heterotrophic nutrition. Fungi only heterotrophic, Plantae only autotrophic, Animalia only heterotrophic. So, amongst all the comparisons you made, which has most diversity and most differences? Nutrition. So, mode of nutrition is the major criteria of 5 kingdom system of classification. Got it right? Everybody? Go through this. If you are having any doubt, you just ask me, and then we will start on 6 kingdom system of classification once. Done? So, here these are the NCERT liners. Monera includes, louder, prokaryotic organisms. Protista is the common ancestor for fungi, Plantae, Animalia. This kingdom Protista has brought together Chlamydomonas and Chlorella, which is earlier placed in what kingdom? Algae, Paramecium and Amoeba, which doesn't have cell wall. Okay. So, which kingdom in 5 kingdom system of classification includes both the organisms which have cell wall and which doesn't have cell wall? Here the kingdom Protista includes both autotrophic organisms, heterotrophic unicellular organisms. That you keep in mind with these examples. Got it everybody? Next. Fungi. What is the new kingdom introduced in 5 kingdom system of classification? Fungi. It has cell wall and it is multicellular, heterotrophic. Plantae has cell wall and this is autotrophic in nutrition. No cell wall, heterotrophic nutrition. Right? Remember this with respect to Protista and fungi. Next. Carl Woese has proposed what system of classification? 6 kingdom system of classification, also known as 3 domain system of classification. 6 kingdom or 3 domain system of classification. So, according to this scientist, there is a common ancestor called as progenote. Okay, what is a common ancestor? Progenote. This progenote has 16S rRNA, and this 16S rRNA is common in all the organisms. All the organisms will have 16S rRNA, and from progenote 3 domains he has classified into archaea, bacteria and eukarya. You may get doubt how archaea and bacteria are separated. It is based on cell wall composition. They are separated based on cell wall composition. So, archaea, if you see, cell wall has what? Polysaccharides and proteins. Cell membrane has branched chain lipids. Only kingdom under archaea domain is archaebacteria. Whereas cell wall of peptidoglycan cell wall of bacteria has what? Peptidoglycan. Only kingdom introduced here is eubacteria. 4 kingdoms under eukarya, what are they? Protista, fungi, Plantae, Animalia. So, this is all about 3 domain 6 kingdom system of classification proposed by Carl Woese. Amongst this, which kingdom has branched chain lipids in the cell membrane? Archaebacteria. Kingdom I asked, archaebacteria. Domain to you have to answer archaea. Done? Yeah. We will start with the first kingdom, that is Monera. All these are the basic things you know. Whichever you find like add-on points or new points, please note it down. These are prokaryotic and they doesn't have nuclear membrane, nucleolus, chromatin. Okay. Histone proteins, membrane bound organelles also absent. Simple. All the prokaryotic cell features we have learned now in Cell the Unit of Life. Same thing. They don't have nuclear membrane, nuclear envelope, nucleolus, chromatin, histone proteins, membrane bound organelles, and they have rigid cell wall. Okay. Monera kingdom organisms will have rigid cell wall. Based on the mode of cell wall, they are again classified into eubacteria, archaebacteria, Mycoplasma, actinomycetes. Eubacteria cell wall is what ma? Peptidoglycan. You are well aware of this. Archaebacteria, polysaccharides and sugars. Okay, Archaebacteria, polysaccharides and sugars. Mycoplasma doesn't have any cell wall. They are the jokers of plant kingdom and they are pleomorphic. They change their morphology or shape based on the host they are invading into. Actinomycetes consists of what? Mycolic acid in their cell wall composition. Okay, very important, please note it down. Based on cell wall composition, they are classified into Eubacteria, Archaebacteria, Mycoplasma and Actinomycetes. And nucleolus, nuclear membrane, nuclear envelope are absent anyhow, but then in the cytoplasm the genetic material is present at a place. Okay, and this region is addressed as what ma? Nucleoid. Nucleoid is also known as what? Incipient nucleus. Nucleoid or incipient nucleus. It consists of double stranded circular DNA, RNA and polyamides. Double stranded circular DNA, polyamides and RNA. Where is your notes? Polyamides are positively charged they will hold on the negatively charged DNA. Done ma? Now a quick recall on the prokaryotic cell where we will understand this easily. When you are revising also, when you are going through the revision of kingdom Monera, you have to read it along what? Prokaryotic cell of cell the unit of life. If you revise that way, it will be a complete concept. So here if we have to start, prokaryotic cell is covered by what? Cell envelope. Has how many layers? 3 layers. Outermost layer is glycocalyx, second is cell wall, third is cell membrane. Glycocalyx can be of capsule and slime layer. And this cell membrane will have what extensions? Tubular, vesicles or lamellar extensions known as what? Mesosomes. Can you tell me the diverse functions of mesosomes we have discussed? Cell wall formation, respiration, secretion, formation of new daughter cells, increase in the surface area. Good. And we have double stranded circular DNA, that is nucleoid, which consists of what? RNA and polyamides too. Along with this genomic DNA, this is addressed as genomic DNA. Along with this genomic DNA, we also have non-genomic DNA, that is known as what? Plasmid DNA. Plasmid DNA has 3 specific functions we have learnt. What are they? Antibiotic resistance. It gives a specific phenotypic character to the bacteria, and it also helps in conjugation. These 3 are the specific functions of what? Plasmid DNA, right? Genomic DNA and non-genomic DNA, clear, no? After which we have learnt on what? The non-membrane bound organelles. Anyhow, membrane bound organelles are absent. So, non-membrane bound organelles includes what? Ribosomes, which are what type? 70S type of ribosomes. Larger subunit is 50S, smaller subunit is 30S. Likewise, we have ribosomes in cytoplasm and attached to the nuclear membrane. The ribosomes which are present in cytoplasm will help in protein synthesis, attached to the cell membrane will help in translocation of proteins. And there is one more phenomena we have learnt in the cytoplasm, that to a single mRNA, many ribosomes will be binded. That condition is known as what? Polysome or polyribosomic condition. Recalled everything? Along with this, we have learnt on one more non-membrane bound organelle, that is inclusion bodies. Rather than saying it as organelle, they are non-membrane bound bodies, storage bodies. Okay. So, with this we have recalled the structure of prokaryotic cell. Now, what they are showing, I already told you this term. They exhibit what diversity? Highest metabolic diversity. Nutrition, so you can see autotrophic, heterotrophic. Again autotrophic, photosynthetic, chemosynthetic. Heterotrophic, we have saprophytic, symbiotic, parasitic mode of nutrition. And then respiration, if you observe, aerobic respiration, anaerobic respiration. Aerobic, you can see obligate aerobes, facultative anaerobes, obligate anaerobes, facultative aerobes. Have to tell you, obligate aerobe or obligate anaerobe means to oxidize the food material, it definitely needs oxygenic environment. It definitely needs oxygen. Facultative, the term sounds easy when you replace this facultative with sometimes. Sometimes it is acting like anaerobe. Facultative anaerobe, sometimes it is acting like anaerobe. Most of the times it is aerobe only. Whenever oxygen condition is not there also, it can perform the, or it can oxidize the food material. Obligate anaerobes, definitely it should require what? Anoxygenic conditions only to oxidize the food. Facultative aerobes, sometimes only it acts like aerobe. Most of the times it is anaerobic only. Understood all? So, these are the 4 different modes of respiration performed by different bacteria. We need to know about the meaning of the term and the examples, enough mark. Okay. Reproduction, 2 types of reproduction we can see. Asexual reproduction, as just we have a single gene or circular DNA, rather than calling it as sexual reproduction, we will call it as sexual recombination or gene recombination here. Okay. Sexual or gene recombination. Likewise, we have what? Conjugation, transformation, transduction. Next are what are? Bacterial appendages. We have 2 types, motile, non-motile. Only motile structure in bacteria is what? Flagella. Non-motile structures include pili and fimbriae. Good. So, now on what basis kingdom Monera is further classified? How the bacteria are classified, you have to know. Whenever you are learning on any classification in biology, you have to know the basis of classification. On what basis? Presence or absence of cell wall, habitat, type of the cell wall. Type of cell wall we have already learned. Archaebacteria will have noted down, no? Polysaccharides and sugars. Mycoplasma, no cell wall. Eubacteria, peptidoglycan. Actinomycetes, mycolic acid. Archaebacteria. These are the most primitive monerans. They occur in extreme harsh habitats. They can occur in extreme harsh habitats because of branched chain lipids that are present in the cell membrane. They can tolerate the highest temperatures. Any adverse temperatures they can survive into. They can be as halophiles in extreme salty areas. They can survive in thermoacidophilic regions in hot springs. They can also survive in marshy areas as methanogens in the gut of ruminants. Okay. So, archaebacteria further classified to what ma? Halophiles, thermoacidophils, methanogens, right. So, here if we see halophils, example include what? Halobacterium and Halococcus. Thermoacidophils examples include Thermoplasma and Sulfolobus. Methanogens, Methanococcus and Methanobacterium. To remember the examples of methanogen and halophile, you cannot struggle much, but you have to remember Sulfolobus, right. This is the only thing where you have to invest your memory on. Rest all you can choose it directly. You will get the PDF. Okay. So, in gut of ruminants, what is the job of these methanogens? Carbon dioxide, methanol and formic acid are turned to what? Methane, which is used for the production of what? Biogas from dung of the animals in the form of dung. Okay, you will get what? Biogas which is rich in methane. Okay, what are the reactants of this? Carbon dioxide, methanol, formic acid, right. Remember the examples, and then eubacteria, this we have learnt. Shapes of the bacteria, and we will stop. Okay. We need to yet do with photoautotrophic, chemoautotrophic examples, types of respiration examples, types of reproduction. This we have to do, and then we will also finish off Protista in the next class. Okay ma? Listen carefully. So, we have different shapes of the bacteria which are categorized to how many? 4 categories based on the shape. If it is spherical like this, cocci, singular form, coccus. Rod shaped, bacilli, singular, bacillus. Spiral shaped, spirilla is the plural, singular, spirillum. Comma shaped, vibrio, plural is vibrio, singular, vibrium. Amongst all this, which is the most pathogenic bacteria? Bacillus, most pathogenic, most common form of bacteria and most pathogenic as well. Right. So, today recap of this class. What did we learn on to? Fundamental insights of this chapter. We have learnt on all the kingdom systems of classification, and we have started with Monera, learned the complete criteria of the Monera, classifications. We are left over with nutrition, respiration, reproduction, useful and harmful bacteria, right. And tomorrow we will also do with Protista.